Alrighty guys, so I'm going to show you how to draw the fluid mosaic model today. So as you can see, I have already drawn in my phospholipid bilayer and labelled my extracellular fluid and cytoplasm. So I'm just going to label the phospholipid bilayer like this. And that consists of our hydrophilic phosphate heads, and our hydrophobic phosphate tails. Oh, sorry, hydrophobic hydrocarbon tails. So the next thing we'll draw in is our integral protein. So that just looks a little something like this. And as you can see, it spans the entirety of the phospholipid bilayer. And then we also have what's called an integral channel protein. So very similar looking, but just has a channel in the middle of it. Just label both of those. The next thing we can draw in is our peripheral proteins. So they just look a little something like this. Sometimes they like to also attach to our integral proteins. So I will label both of those. And the next thing we can get is what's called a glycoprotein. So it looks a little something like this. So it basically just has a carbohydrate group attached to the protein. And that is our glycoprotein. And we can also get what's called a glycolipid. So similar to the glycoprotein, just a carbohydrate group attached to a phospholipid. And then last but not least, we can get our cholesterol, which sits in between our little phospholipids here. So I'm just going to represent that with some green squiggly lines. So that is our cholesterol. And that is how we draw the fluid mosaic model.